Well, good afternoon, good morning to all of our clean slate employees in Canada and the USA, and also a special welcome today to our external collaborators and friends who are joining this presentation today. On behalf of Dr. Carolina Koutras, our Director of Clinical Research at Clean Slate UV, I'm your host today, Jeff Cole. I'm Director of Engineering. So thanks to everybody who are joining us today to explore how engineering solutions can help prevent the spread of pathogens in healthcare and beyond. Our speaker series hosts engineering and health leaders to discuss new research and innovation in the area. To kick off our series in 2021, we are pleased to have Dr. Ben Hatton from the University of Toronto join us to present his research on surface contamination and the design of antimicrobial materials. Dr. Hatton is an associate professor at University of Toronto, where he leads the Functional and Adaptive Surfaces group in the Department of Materials Science and Engineering, and is also cross-appointed to Biomedical Engineering and Mechanical and Industrial Engineering. Dr. Hatton's training started with a bachelor's degree at Queen's University, master's degree from McMaster, and his Ph.D. at University of Toronto. He did postdoc work at the National Institute for Material Science in Tsukuba, Japan, and then at Bell Labs, Murray Hill, New Jersey. He subsequently held research associate positions at Harvard University and the Wyss Institute for Biologically Inspired Engineering before returning to Toronto in 2012 to establish the Hatton Lab. The Hatton Lab group currently studies bio-inspired materials design, surface wetting and adhesion, biological interactions with material surfaces, particularly microbial contamination of biomaterials, antimicrobial materials design, and biomedical devices. So with that said, welcome Dr. Hatton. Please take it away. Thank you.
Great, thanks so much, Cole, for that introduction, and thanks very much for inviting me to give a talk. Unfortunately, now I'm having trouble sharing my screen. It worked before, but when I click on the link, it doesn't give me the option of choosing. Yeah, I don't know what to do here, actually, sorry. It worked in the test, right? I guess you can't see anything, right?
Not yet.
Well, this is frustrating.
Link.
Yeah, when I click on share your screen, it just doesn't give me the option anymore. I don't know why.
Okay, strange. Let me see if I can add you here. Would you like to send me your presentation and I can share it and I can switch slides? That's strange.
I think that would be tricky. Maybe if I leave and join again or something, or?
Yes,
Yeah,
maybe try rejoining with the link that I sent you in the last email.
Yeah, like the actual link, right? Yeah, I'll try that again. So why don't I try
Okay.
leaving?
We'll give him another minute.
For those of you that have joined us in the last few minutes, today's presentation, it will be by Dr. Ben Hatton of the Hatton Lab at University of Toronto.
There you go.
Okay. I can't seem to pick the window, but I think I can work with this because you can see that, I hope. Can you see the whole screen?
Yes, yes,
Okay, great. Okay, so I think I'm ready to get started then. Well, sorry for that hiccup. It always happens, right? Works perfectly when you test it. Okay, so again, I'm Ben Hatton. I'm an associate professor in the Department of Material Science and Engineering at the University of Toronto, and I I run a group called the Functional Adaptive Surfaces Group. And today I thought I'd talk about the role of topography and surface damage on microbial contamination of those materials. And how do we design antimicrobial materials? So basically, how does material science come into this problem of microbial contamination? So this is my group here. We're about sort of 10, 12 people. And we sort of cross this boundary between the design of materials, our fabrication lab, and our microbiology lab. I think you can see my pointer, right, when I move it around. Is that right? So we're able to grow bacteria with a level 2 facility, and we can design materials. I'm interested in material surfaces because so many properties of materials really depend on just the surface, actually, quite independent of the bulk properties of the material. So things like, well, biofouling I'll be talking about today, cell interactions with surfaces, but also adhesion, wettability, friction, all these things just depend on surface properties. And over the past years, sort of 10, 12 years or so, I've been really looking at sticky problems. So how do things accumulate on material surfaces, like ice, for example? Anti-ice surfaces is something I've worked on for a while. Marine biofouling is something I've worked on. I'll be talking about bacteria today, but also blood clotting. We've worked on that. So these are tricky problems. How do you prevent these things from adhering to material surfaces? And this is sort of an overview of what we do in my group. So we have sort of a few major areas. One is in sort of porous material layer design, but also in this second project here, we do a lot of stuff with designing surface nano and micro topographies to control wettability and adhesion. This is what we're talking about today, especially bacteria growing on material surfaces. And then we also have a whole extra project basically on active surfaces where we can actually move topographies around. So bacteria on surfaces. This is quite a big sort of complex story, right? It really depends on what environment and context we're talking about. So this is a problem for medical devices, like implanted medical devices in the body, where it's obviously a wet environment and can be quite long-term for certain devices. But there's also situations like fomites, right? Touch surfaces like door handles and so on, associated with infectious disease transmission. Obviously, that's a big deal these days. But this is sort of a dry kind of environment, right? It's outside in the air and kind of can be short-term. There's a problem of bacteria contaminating surfaces in food processing environments, can be sort of a wet environment, sometimes sort of wet, dry. And then there's things like biofilms in drains. This is a big problem in hospitals where it's really a wet environment typically. So these are all different types of contexts and environments where bacteria live on material surfaces. And we've actually worked on sort of all of these different types of problems. So bacteria, when they have a chance to grow on a surface in a wet environment, they grow bacterial biofilms, and these are protected by this extracellular matrix that protects the colony, and eventually they redisperse and find new surfaces to live on. Biofilms grow on medical devices. Things like the central line catheters or these endotracheal tubes always develop microbial biofilms. And it's just a question of whether that leads to a body infection. And so materials are key to this process, actually. We're learning a lot more about it. But if you sort of see all the different surfaces that bacteria can colonize, all these new fresh surfaces that are available are perfect for contamination. And these hospital-acquired infections are, in fact, a huge problem, very costly problem. And in fact, they're the 6th leading cause of death in the US, ahead of even diabetes, influenza, and Alzheimer's. So huge costs, and it's a very serious medical problem, complication that tends to develop, right? And we know certain strains that are associated with certain kinds of devices and so on. And 60% of these are associated with medical devices. Within the hospital, you also have a problem, as I mentioned, of drains. So within this ICU, the drain is in fact the only part of this room that we really cannot disinfect properly. So as you run the sink, it sprays an aerosol in the room, and it's because of this colony, biofilm colony in the drain that is living there all the time. It's a huge problem. So we work with a group at Mount Sinai Hospital to sort of put non-fouling surfaces on their drains. But I won't talk about that too much today. Another problem, as I said, is food processing surfaces. We have conveyor belts, massive surfaces, very hard to remove and disinfect bacteria completely from these complex environments, leading to food outbreaks sometimes. So bacterial interactions with surfaces is quite a complex relationship. There's a lot of mechanisms involved and factors. So it can be electrostatic interactions, sort of physical attachment interactions. And certainly an important part of this is the conditioning layer that bacteria put on a surface. This can be protein or polysaccharide that enables them to attach to a material surface. And topography, as I'll talk about, is another really interesting and kind of complicating factor that determines how well bacteria can attach to surfaces. Here's an example of a conditioning layer. This is a quite well understood mechanism for Pseudomonas aeruginosa. That's a good example. These red stains here are trails of this polysaccharide cut PSL that they deposit that enables them to attach and grow a biofilm on the material surface. So this, we can stain for that PSL and see exactly where they're putting down this conditioning layer. I'll talk more about that in a minute. So within hospitals, pathogens can survive on material surfaces for an incredibly long time. We don't really understand the factors that control this. But when we look at certain pathogens like C. difficile, Enterococcus, Pseudomonas aeruginosa, and Staphylococcus, including MRSA, this can range anywhere from, hours, but all the way up to months. And we don't really understand what controls that, to be honest. Like, what is it about the material, the environment, the humidity, and so on. But certainly topography is a factor. So here you can see a groove or roughness on the surface of stainless steel. That's where bacteria are growing. And we know that roughness and topography, essentially surface defects like grooves and scratches, give that sort of initial attachment site for bacteria. So disordered roughness generally increases the amount of bacterial attachment. So when you've got medical implants with some roughness, it's those defects that give them the chance to attach to the surface. And that's sort of well understood over the years. And we can see that ourselves. This is some work we did with silicone. We work a lot with silicone. So this is PDMS silicone, where we took a nice, fresh, clean silicone sample and we gave it various cycles of autoclaving to progressively damage the surface. And you can see these sort of scratches and grooves and roughness that's developing as we go through 10 cycles of autoclaving. And we've, in this case, grown bacteria at every stage prior to autoclaving. And we can see that the amount of bacterial growth is increasing with that damage. So at each stage, we're growing new bacteria, but the amount you can see in the crystal violet stain is increasing. And you can actually see in this fluorescent stain of the bacteria exactly where they're following these grooves and defects on the surface. We can even take a piece of silicone and sort of stamp it, emboss it with these little micro cracks. These are about 10 microns apart, these cracks here. And you can see the bacteria, in this case Pseudomonas, are exactly following those micro cracks. So they use those defect sites as their initial site for attachment. And to sort of make that point even more clear, we did an experiment recently where we took several pieces of PDMS silicone and we bent them slightly. So one side has a slight tensile stress, one side has a slight compressive stress. And I'm just going to check on things here, okay. And allow the bacteria to choose which side they want to grow on. They can choose quite easily either side. They have full choice there. And there's the experiment there. And what we find is if you have new pristine silicone, then the bacteria don't have any preference. They'll choose one side the same as the other. So whether it's unbent or bent, if it's new silicone, perfectly new, pristine, then there's no difference. But if you wipe that silicone, just wipe it with a paper towel a few times, all of a sudden you see that the bacteria in the bent sample here choose the tensile side of the sample and do not so much like the compressive side. So it's like 3 times more on the tensile side than the compressive side. So why is that? Just wiping a sample and bending it is giving them this preference, right? So they're choosing the tensile side. Well, just wiping the sample, as it turns out, with paper towel is enough to put these little, very small micro cracks on the surface. So this is a surface profilometry work that we've done you can see the damage that's there. This is that red dotted line in a profile, much, much bigger than the pristine and the unbent, so the compressed sample. So on the tensile side, we're seeing these micro cracks more prominent than on the compressive side. Essentially what's happening is the tensile side is opening up those micro cracks and making them much more available than on the compressive side, which is actually closing them down. And if you look at the fluorescence, you can see where they're following these defects on the surface, these micro cracks. And on this tensile side, they have a much bigger variety of defects to choose from than on the compressive side. So what does this tell us? Well, first of all, micro damage can occur very easily on polymer materials, especially elastomers like silicone. Even just wiping it or handling it is enough to put these little defects in the surface at the scale of the bacteria. They're just so small. And that's not something you would notice. It looks fine. And if you bend the sample, you're opening up the defects on the tensile side a lot more than the compressive side. Okay, so it shows us that they're very sensitive to defects and they prefer to colonize those defect sites. How do we design to prevent that, right? So typically antimicrobial materials are loaded with antimicrobial species, and this is, sort of effective. So silver, chlorhexidine, triclosan, these are common things that will diffuse out to the surface and kill bacteria that are on the material surface. And this is semi-effective, but it depends on that diffusional release. It takes time. And very often bacteria start developing a resistance to that. But the thing is, it doesn't stop bacteria from adhering to that surface. And so bacteria start growing on the dead cells that are there. And also it needs a wet environment, it needs a solvent typically to migrate to the surface. So it's not so good for short-term and dry fomite conditions. So we prefer approaches that are kind of bio-inspired that actually prevent bacterial adhesion to the surface in the first place. And these can be chemistry approaches, at least sort of this one here, where we degrade the matrix of the biofilm, or these can be topography approaches that are affecting the wettability of the surface. And so these are all approaches that we've worked on. This first one is sort of antimicrobial drug release. This one here, I'm going to talk about briefly. This is putting enzymes on the surface to degrade the polysaccharide. And then of these 3 topography effects, this approach 3 here is where you have a very small scale nanotopography that some people have shown can kind of physically damage the cells. We're not such a believer in that approach, but we have done some testing of that. But I'm not going to talk about that today. I'm going to talk about this approach, non-wetting super hydrophobic surfaces that use a sort of a non-wetting topography to limit bacterial attachment to the surface, and these slippery surfaces. So the last one I'm going to talk about is this project on slippery material design. Okay, so if I keep track of the time here, I should have time, I think, to talk about those 3 projects. I'm just going to check the time here. Okay. All right, so the first project we're going to talk about is this biofilm degradation. This is work we do with sick kids where we actually we sort of disrupt this biofilm formation process. So again, bacteria form these matrix sort of protecting biofilms on a material surface. But at some point, inherently, the bacteria have to produce something that actually degrades that matrix so that they can escape and find new surfaces. So what this group at SickKids has done, this is Lynn Howell who leads this group, they've found that certain enzymes can degrade the matrix of biofilms and make them very susceptible to antimicrobials. So what we've done, I can speak more about that, but I'm just going to sort of go through quickly. What we've done is interfere with that process. And so whereas bacteria normally put down this PSL polysaccharide as a conditioning layer that enables them to attach to a material surface, what we've done is taken these special enzymes, these glycoside hydrolase enzymes, and attach them to our material surface so that they just continuously degrade this PSL polysaccharide. And in fact, they do it so effectively, continuously, that they prevent it from absorbing to the surface and the bacteria simply can't attach. So this works very well for specific strains, in this case, Pseudomonas aeruginosa. So here's sort of the chemistry of how we've attached these PSLG enzymes to the material surface. I won't go through the details of this, but I can talk about it more later. And we've shown that the enzymes are on the material surface by different means. And what we can see is that over a period of 8 days in continuous culture, compared to sort of normal glass control surfaces, our enzyme modified glass really is significantly reducing the amount of bacteria. I think I had the numbers there, but certainly by a factor of sort of log 3 to log 4. So I forgot to put the CFU numbers there. And we can see that happening in situ even. This is a quartz crystal microbalance that measures the absorption of species to material surface, and this blue curve is the normal curve showing how PSL absorbs onto the surface, whereas our red curve is showing that there's really nothing absorbing. So these enzymes are just totally constantly degrading the polysaccharide. And where you put the enzyme, you get very few bacteria. So we can pattern the enzyme on the surface and show that it's keeping those bacteria from attaching locally. So I'll go through that quickly, but that's sort of a project that's ongoing, that's specific to Pseudomonas. But the other two projects I'm going to talk about are really non-specific because they're kind of physical means of preventing bacterial attachment to surfaces. And so the first one of those are super hydrophobic surfaces. So these are surfaces that have a topography, a nano or microscale topography, which is also hydrophobic. And when you combine a topography, a roughness, with a hydrophobic surface chemistry, you get what's called this. This is the lotus effect. So this is, many plants have this property where they can cause water droplets just to bead up and roll around. And the lotus plant is famous for this. And we think that it's for sort of a self-cleaning for plants. So water droplets sort of roll around and pick up dust on the surface. And what's happening here is the hydrophobic plus topography is making the liquid interface of this water droplet only stay on the tops of the features. That's how this lotus effect works. It keeps all of the liquid of the water droplet just touching the tops, right? And that's what's called the Cassie state. So the water droplet is just sitting on the very tops of those features. And plants have beautiful complex structures on their leaf surfaces. Not all of them are super hydrophobic, but many of them are, with grooves and little posts and wrinkles and things. So what you might think is, well, if the liquid is just stuck to the sitting, only on the tips of these features, that can limit bacteria from attaching. And that's what sort of people have tried over the years. I've also done work on sort of limiting that contact for anti-ice surfaces and so on. But when you try it with bacteria, you find it doesn't necessarily work. for various reasons. But one factor that we found was important is actually the size of the surface features. So generally, if you have features that are much bigger than the cells, you can have a condition where the cell features about the same size as the cells, or where the features are much smaller than the cells. So what's the ratio of the feature size to the cell size? That's what we want to test here. And of course, if we can limit the bacteria to the tops of these features, and then we can presumably limit how much they attach to a material surface. So hopefully that's clear. What we've done here is test 4 different strains of microbes: Candida albicans, a fungus, yeast, E. coli, Pseudomonas aeruginosa, and Staph aureus. All have different sizes of cells, slightly different. And we've tested material surfaces that have a widely ranging size of surface topography. So going from a flat control to very large posts. These are 150 micron diameter posts. We've got a few in between here, down to sort of 5 micron, 3 micron size posts, so 1.5, and down to 0.3 micrometers, so 300 nanometer size posts. But all of these post surfaces have about the same area fraction of posts. It's just that we're going from fewer number of large posts to a very large number of small posts. And all of these surfaces, well, the ones with the posts, are all super hydrophobic. So you can see the water droplet is kind of beaded up here. So in each case, they're just touching the tops of the posts. Okay, so we've run bacteria for a relatively short time, on the order of sort of a few minutes to sort of 30 minutes. And what we can see is that on a flat surface, this is polyurethane, you can see all the cells there, in this case E. coli, at a relatively high density. When the posts are big, in this case these are 20 micron posts, you can see them all sitting on the tops of the posts, and they have no problem attaching there. And actually the cell density is still pretty high, even though they're just limited to the tops of the posts. But as we get down to smaller posts, 3 micron posts, the density is coming down a bit. And at a certain point, at about 1.5 and smaller, there's very, very few cells attaching anymore. And so you make the post small enough and they have trouble attaching or staying on, right? And there's the 300 nanometer post. This one's molded in polyurethane. You can see the flexible post there. And this applies for all of the microbes we've looked at. So Candida, Staph aureus, Pseudomonas, and E. coli all show significantly decreased numbers as you get to very, very small topographies, small feature size. And there's the numbers. It's incredible. So we're seeing for Staph aureus, for Pseudomonas, for E. coli, and Candida, an enormous change in the number of cells that are attached at the large post size or the flat. They're actually quite similar. If you see for Staph aureus here, there's the flat control. There's the 150 micron posts, and it goes progressively down as you get to smaller and smaller features, down to, let's say, 0.3 microns here. It's on an order of like a log 4. So 10,000 times difference between the large posts or the flat surface down to these small nano posts. And we see that same trend for Pseudomonas and for E. coli and for Candida. So there's a 10,000 times reduction just by making the post small. What's interesting, I could talk a lot more about this, but it also depends on the size of the cell. So Candida is a much bigger cell compared to Staph aureus, so everything is actually shifted a little bit over. So even 1.5 micron posts are small enough to keep the Candida off the surface. So this is a sort of project still we're kind of working on quite a lot. We're actually interested in modifying medical gloves with these types of surfaces so that as you touch contaminated droplets or wet materials, we're not going to be picking up so many microbes with every contact. And we think this is actually a surface tension effect that's keeping the bacteria from attaching to that material surface. Okay, so topography can have a big effect to limit bacterial attachment under certain conditions, whether it has to be non-wetting and it has to be small enough features. That's kind of the conclusion we've got there. And we have a paper just about ready to put in now. Okay, hopefully you guys are still with me. I can't see anything here, but I assume you're still there. So that brings me to the last project I'm going to talk about, which is this design of slippery surfaces. So this is the last approach. This is work that I started with a group when I was at Harvard. I was at Harvard for about 5 years, and a friend of mine actually came up with this idea. And it turns out this to make slippery materials. And this is in fact what happens in nature quite often. So a good example of this is the pitcher plant. This is a carnivorous plant that has a very slippery edge here, just on the edge of its sort of carnivorous, extended leaf thing that traps insects inside. But it's only slippery under dry conditions. It's under wet conditions. Under dry conditions, insects can crawl all over the edge of this leaf structure, no problem. But under wet conditions, they fall inside. And what's actually happening here, this is a video that illustrates this change. So under dry conditions, you can see, hopefully, the ants crawling all over the top of this pitcher plant. But as soon as it's humid and wet outside, they just slip right in. They have very little resistance to hold onto that surface. So what's happening here? Well, apparently, or what's actually happening through various sort of studies is that the plant actually traps water. It's a hydrophilic surface that's trapping water into these microgroove structures on the plant edge. And so you can see the water, this is frozen to prove that it's there. And the edge of this pitcher plant is kind of devious because it's very slippery here. The insects fall in and they can't get out again. So that thin water layer is actually a barrier to them getting adhesion to the material surface. So my friend Tex-Sing Wong sort of generalized that idea to say, if we've got a microstructure topography, and we can trap a thin layer of a lubricant liquid into that surface topography, then we can actually essentially have a very smooth, slippery liquid interface at a solid material surface. And that's what we call slips, slippery liquid infused porous surfaces. So Tex-Sing sort of came up with this idea, and now he's at Penn State, working on lots of aspects of this, but this was our first paper on this that sort of generalized that idea. And it sort of really turned into quite a successful principle, a design principle. The key is you make that lubricant liquid immiscible with its surroundings, and now you have a very slippery material. So if we trap that liquid interface there, it's stable against pressure. It's actually thermodynamically stable. It's stable against damage. If you sort of scratch it, it will just flow back and heal that damage. You can choose a lubricant liquid that's chemically inert, and it essentially becomes topographically flat, right? That interface is perfectly flat and slippery. So chemically and physically, bacteria, for example, cannot absorb or attach to the material surface. They can't produce a conditioning layer that will absorb to the material surface, and they have no physical means of attachment. So I won't go into the details of this too much, but it's really based on this selective wettability. So if we trap this lubricant, liquid B here, the blue color, into that surface topography and choose it such that it wets the surface preferentially compared to anything else, in this case liquid A. So if we choose this case where we have this stable condition where the liquid A cannot displace the lubricant liquid for that material, then you have a perfectly stable thermodynamic condition where we want to avoid this condition here where the pink liquid will displace it. And so that lets us design a material surface and choose a lubricant liquid that will enable this. And that's really what we've been doing for the past 10 years or so, and various other people as well. And over the years, we've done various different examples of this. I won't go through them all, but this is work by my friend Nikki Vogel, where we generated these in a transparent layer. So there's in this glass slide here, there's a transparent surface porous coating with a trapped lubricant. And this is a fluorinated liquid that won't allow the wetting of other things. So in the case of water, octane, or even blood, you can see that it won't allow adsorption of those species. And this is ketchup that's de-wetting from that pattern surface there. And Nikki also patterned these sort of H patterns on it to show that you can actually get things like octane and blood to de-wet from these slippery regions. And designing slippery surfaces is very much a sort of biological approach, right? So the way that biological organisms have kind of slippery or mucus covered surfaces, such as within, in the body, various kind of mucus layers that we have inside the intestine, for example, is an interesting way to prevent, well, I guess we're still understanding it, but preventing infection and preventing damage. And there's various examples where sort of surface fouling is really one of probably the key design objectives for having that mucus layer. So how do we apply that idea to various situations where we need to prevent bacterial attachment? There's a lot of examples that we've had over the years, but this is a couple I'm going to talk about here. One is the food processing environment, where we have a problem of bacteria getting into the scratches and grooves on materials like stainless steel. And once they get into those grooves, they sort of form local biofilm colonies, and it's very hard to get rid of them. It's hard to disinfect. So in this project, we took stainless steel, so standard sort of rough-ish stainless steel, this standard one here, and also one that was very, very smooth, so mirror finish. And we measured the surface roughness of those. You can see the profile of the topography cross-section. So it shows you all of these little micro grooves and damage that's at the surface. There you can see sort of examples of those topography, the roughness, and the bacteria, in this case E. coli, that's growing on these material surfaces. And again, they tend to seek out the damaged sites, the grooves and the scratches on the surface. What we wanted to test is whether, this is the standard condition. So stainless steel, you can see all the grooves and structures and the bacteria and the residue, organic residue that's getting into those grooves. We wanted to test whether making the surface hydrophobic and putting a layer of oil, just regular cooking oil, mineral oil, corn oil, olive oil, we've tried lots of things like that. So it's food safe, and we're just putting a layer of oil onto that hydrophobic steel, all right? And in fact, if you do that, you find that the oil is, now preventing bacteria from attaching to that metal surface. That's a bit naive, though. In fact, what we really wanted to test is what happens after you kind of wear away that oil. Like if you've got an excess oily surface, that's sort of one thing. But as you wear away the oil, we wanted to see if it could really block just those problem sites, the defects on the surface. So filling in those grooves and scratches to find this kind of condition. And in fact, we found that this can work really very well. So here you can see the regular sort of standard stainless steel in the 4-hour and 24-hour condition of growing E. coli. With excess oil, we see almost no bacteria, actually. It's very, very effective. But even after significant wear of that oil layer, we can see that there's, in fluorescence, at least in these examples, you can see actually no bacteria. When we do a colony forming unit count, we can see that compared to the uncoated case in 4-hour growth, well, there's actually none detected when there's excess oil. There's actually no bacteria. After wear, you can see that it's sort of 2.5 log reduction. So sort of between 100 and 1,000 times reduction then. So that's very significant just for having a little bit of oil on the surface, right? After 24 hours, you can see that it's well, 7 to, so it's, but still sort of 2.5 log reduction after wear. So that's a very significant change in the contamination of surfaces just from having a bit of cooking oil. So that was actually quite interesting. And it's blocking the defects. That's really the key idea here. And we see that also for the mirror finish steel, but it's less of an effect because there's fewer surface defects. So there you can see kind of the oil surface and the standard surface. In this case, we just wanted to show that the surface is also much easier to clean. So wiping away microbes and contaminants is much easier when you have even a tiny amount of oil at the surface. Okay, and sort of the last part of this section on slippery surfaces is what we call self-lubricated elastomers. So this is a project I initiated at Harvard to take regular silicone, so PDMS silicone, and what we do here is infuse it with excess silicone oil. And what that does to the silicone rubber or elastomer is it makes it swell. So it takes up the silicone oil, but that silicone oil isn't bound to anything. So it's just sort of making the polymer swell. And what it does, in fact, is it migrates to the surface. So that's why we call these self-lubricated materials. So it actually forms a very, very thin layer of silicone liquid at the silicone rubber surface. So it feels sort of slippery. And what we wanted to test is whether that really thin oil layer can prevent bacteria from attaching. And in fact, it works amazingly well. We've tracked how rapidly the silicone oil is migrating to the surface. This is atomic force microscopy images where, again, I showed you that example where we patterned microcracks in the surface of silicone. So that's what these lines are. And we can see the silicone oil migrating up through into those microcracks over time. And this was an early example of what we did here. This is a piece of silicone tubing where this half of the silicone was swollen in this excess silicone oil, and this half was not. This is the untreated side. And this is crystal violet dye. So you can see clearly that this side is growing significant numbers of bacteria, and this side has very little. Here you can see how the silicone tubing swells in the presence of silicone oil. And recently what we've done is test this quite extensively. This is a 30-day culture for Pseudomonas aeruginosa, a continuous culture. So very, very difficult to keep bacteria from growing on these surfaces for 30 days, believe me. So in the top row here, we can see the untreated control, so just regular silicone. It's accumulating biofilm growth very, very quickly within a day even. So 30 days, you have extensive biofilm growth. But the silicone slip surface, so the slippery self-lubricated silicone is this bottom row, and you see, well, really up until 30 days. Here you see a little bit of stuff coming in from the edge of the sample, but I think that's just an edge effect, to be honest. So we see a very, very stable resistance to bacteria growing on this material, really because of that very thin silicone oil layer. So here you can see the colony forming unit counts over a period of 1 to 30 days. And again, we're seeing 10 to 3 to 10 to the 4, so 1,000 times to 10,000 times reduction in cell density over that 30-day culture, which I think is really astounding. As we add topography to those materials, we can see the silicone oil layer filling in the grooves. This is a sort of a grooved pattern that we put into it. We can see over time that the oil is kind of coming up through the bottom of those grooves, which kind of leads us to an important point here. If we now have a damaged silicone, right? I showed you the example of wiping the silicone or putting the microcracks in. What we can see, in fact, is the silicone oil is a self-lubricating migration. It comes in and fills in those damaged sites. It fills in the cracks and the grooves and the scratches that are at the surface, and it conceals them from the bacteria. Those sites are no longer sort of preferential sites for attachment. And so as we take PDMS silicone, here's the sort of pristine material. This is wiped, again, with the paper towel. We can see all the little grooves and scratches that are there. You can see the size of the scratches in this AFM profile. This is embossed with micro cracks, so you can see the clearly spaced cracks in the surface. And over time, this is on the sequence on the right here, you can see how the lubricant comes in, fills in those cracks, and you can't see them anymore. After several hours, it conceals those cracks completely. It just looks flat now. And if you then give this sort of these different materials for bacteria to grow on, here's just the regular sort of flat PDMS, you know, wiped condition, and there you can see the damage that occurs in these groove structures. And over time, you can see how the bacteria are able to attach to the surface when there's no lubricant there. And in this bottom example here, we can see that when there's lubricant present, it's keeping bacteria from finding those micro cracks. So you can see no evidence here. You can see they're clearly following the micro cracks, but in this case, they're simply not there. Okay, so that sort of is quickly sort of the different projects that we've had to look at how topography can affect microbial attachment to surfaces, and how even a small amount of damage, even wiping or bending silicones is enough to sort of put in these microscale defects. But how certain approaches that we can take can actually inhibit that initial attachment of bacteria to surfaces. So in that first project, it was putting these glycoside hydrolase enzymes to continuously degrade this polysaccharide glue that bacteria use to attach to the surface. And these two other projects, sorry, I forgot to number them properly there. One is this sort of effect of non-wetting topographies, these microporous surfaces that limit bacterial attachment, but only when they're small enough. And then all of these different examples of slippery surfaces that provide a kind of a liquid interface at the material surface that prevents bacterial attachment, even over long periods of time. But particularly in this last case, it's really due to blocking or concealing these defect sites that it seems to be effective. So I want to thank everyone who's contributed to this work from my group, particularly Dalal Asker, our resident microbiologist, our collaborators at Sick Kids Research, the group of Lynn Howell, and various researchers from my past at Harvard, including Joanna Eisenberg, Texeng Wong, and Phil Kim, and funding from our various supporters. So thanks very much, and I'm happy to entertain any questions you might have. Maybe I'll stop sharing.
All right. Well, thank you, Dr. Hatton, for this excellent and insightful presentation. Wow, I sure learned a lot of things. So at this time, do we have some questions? Dr. Koutras, what's going to be our protocol on questions? Will mics be open, or should we have questions in the chat? Well, I see one question in the chat here. Can we apply this material not only gloves, but also to the gown of the healthcare workers?
The gown, absolutely. That actually is a really interesting question, because actually about 3 years ago, we actually got really interested in textile design. So textiles are interesting because, of course, you have a topography, right? You've got the weave structure of the fibers giving you kind of a really microscale roughness and topography. So if you choose a hydrophobic textile and you have that kind of weave structure, you can actually get very good super hydrophobic fabrics. And you can actually see that on some kinds of sports clothing and stuff. When water gets on it, it just sort of beads and rolls away, right? There's even like a spray, you can spray your pants and stuff. There's YouTube videos of that. So we actually tested some super hydrophobic fabrics, and it can be very effective, yes. The only thing we're not sure about is, so we saw a similar result actually to our kind of microposts, in fact. Not quite as good, but actually really can be quite good. So a factor of 10 to the, so log 2 to log 3 kind of reduction. But what's not clear is whether, if you, let's say you applied that to kind of medical scrubs or something, washing cycles, I think, might just kill that effect over time. So they can be a little sensitive to damage and wear, unfortunately. So medical gloves are kind of our favorite thing because they're disposable. You know.
All right. Next question is, with more people conscious about hygiene and the potential presence of harmful bacteria on our everyday objects, do you think it's important for objects outside of hospital settings, such as phones, laptops, cars, etc., to adopt antimicrobial surfaces?
Yeah, absolutely. And this is where things get difficult, actually, because slippery surfaces like we've been working on will only work in a certain context, right? It'll work maybe within indwelling medical devices in wet environments, but it's not going to work on door handles, right? Because you have to hold on to the door handle for it to work. So some of these will work in certain contexts and not in others. So when you've got sort of dry fomite conditions, it's actually very difficult to prevent microbial attachment. This is where I think silver and copper can be much, much better, in fact, because they're constantly releasing metal ions and killing the bacteria. And of course, you know, UV disinfection methods, I think, are a very good non-specific way to kill bacteria on material surfaces. So all these different approaches to antimicrobial, you know, materials, it very much is context dependent. So, but yeah, we're very interested in sort of that infectious disease spread throughout hospital environments and how can we best make materials and treatments to limit that infectious spread. There's another question there I can see. What's the difference between copper and silver antimicrobials and slips? Well, that's a great question. Like copper and silver certainly have their place. And as I said, I think those are best used when you've got a sort of a dry fomite condition, so door handles and things. And the copper and the silver, well, silver is antimicrobial very effectively. We still use it in wound dressings and release from certain medical devices. And copper as well. They both seem to interfere with the replication of bacteria, and they're generally non-toxic to people. So, but those diffusional methods they're a little bit limited when you've got a long-term pressure. Like bacterial biofilms will eventually overwhelm it because the new cells just grow on the dead cells, and you still get growth. But it can be good for killing bacteria. Shall I keep going here with some of these questions, or?
Sure, why not?
Yeah. So I see another question. Do we risk erosion of small posts by frequent cleaning? Well, this is one of the challenges. Yeah, as we put these kind of like sort of small scale topographies on surfaces, we do potentially risk them being damaged by physical damage or erosion. Absolutely. Our approach to that is to, first of all, look to things more stable than just microposts. I mean, microposts are beautiful, but they're not so strong. So for example, groove structures, like those pyramidal shaped kind of groove structures, are much more stable. We're testing those now. And well, when we think about these topographies, like that non-wetting topography effect, the super hydrophobic effect, I really do think it is best in sort of short-term conditions, actually, because it doesn't last forever. The effect actually doesn't last forever. If you hold it underwater, eventually they'll wet after about an hour or so. And so that's why temporary sort of disposable devices like medical gloves are probably the best there, because they're not going to last forever. There's another question. What about C. difficile and other spore-forming bacteria? Well, that's a great question. In fact, we've done some work with Candida, which is a fungus, and all of these, when we think about these physical effects about attachment, like for example, to the non-wetting topographies, certainly the size of the spores and the cells is a big factor. So we're seeing a big difference between large yeast cells like Candida and small cells like Staph aureus. And spores, well, bacterial spores can be quite small, but I guess not that much smaller than the cell itself. But it's a good question. We have to test that a bit more. We're even sort of thinking about viruses, which are even smaller. For the slippery surfaces, well, they're interesting because that's a very kind of non-specific absorption effect. When you have this liquid interface at a solid surface, proteins can't absorb, polysaccharides can't absorb, so everything just kind of slips away. So that would apply to spores as much as anything else. At least that's what we think. Yeah. Another question is, how does material degradation over time impact surface susceptibility to microbial adhesion? And is there an easy way to test for this? So degradation, yeah, I mean, long-term effects are certainly an important thing for our approaches. The long-term effects of, say, the enzyme modified surfaces, the degradation of those, degradation of the non-wetting topographies, and degradation of the slippery surfaces. Well, the enzymes, we've done now a several week study of continuous culture, and the enzyme surfaces, like these enzymes in particular, are really quite stable. Even when we sort of store them long-term, they seem to be quite stable. But eventually they will degrade, presumably. They're gonna, we have them covalently bound to the material, but something will gradually chemically degrade that, I guess. But in that case, if we can get it to last 30 days, that's actually pretty effective, let's say for urinary catheters or endotracheal tubes. Microscale topographies, yeah, we don't actually expect those to last too long. As I said, they'll get physically damaged over time. And that effect, that lotus effect, is sort of a bit time dependent. But the slippery surfaces, actually, they have the potential to last a really long time because it's a thermodynamically stable state. And in fact, we've been a part of a couple of marine fouling experiments. I used to do that down in New Hampshire, and we've done some here for zebra mussels in Ontario. And these materials are very stable, even under these kind of aggressive marine environments. So I think that's our most stable approach. I think I got that question mostly, right? There's another question here. Would bacteria be able to develop the ability to attach to low adhesion surfaces after repeated exposure? That's an interesting point. So can they sort of learn how to adhere, right? That's quite possible. When we're talking about the non-wetting topographies, like the microporous and things, what's sort of interesting is we're looking at a size effect, right? So the size of the cell compared to the size of the post, right? What we're wondering about is when bacteria sort of group together, they can maybe sort of bridge several posts at once. And so Staph aureus, for example, kind of grows in clusters, right? So they're effectively like a larger cell. That's a possibility. I guess we have to test a bit more about, you know, how bacteria might change the way they grow to attach to a surface. Yeah, it's a good question. But since it's a physical effect, I think it's difficult for bacteria to really adapt, you know, but we'll see.
1 o'clock. How about one last question for the group?
Oh, sure. Okay, so there's one more here that says, when microbes slip off, are they still living and how do you dispose of them? Yeah, absolutely. So how do you deal with these bacteria that are not attaching to the material surface, but they're still alive, right? And that's typically what we have. We're not killing them, we're just preventing them attaching. Well, in fact, when you think about medical device contamination, keeping the bacteria off the medical device is huge. Like that's like, you're really more than halfway there if you can keep them off, because when bacteria are free swimming, like planktonic, they're much more susceptible to first of all, antimicrobials, but also our immune system. So bacteria hate being planktonic, it seems. They like being in these kind of protected colonies like biofilms. So that seems to be what we've seen too. When we give these bacteria exposure to antimicrobials, when they're not allowed to form a biofilm, we can get rid of them really quite easily. And Lynn Howell has shown that also for her therapies. And so keeping them off the surface is really important, because that's what bacteria really are trying to do, is live on a material surface. Okay.
We've come to 1 o'clock. Dr. Hatton, thank you again. It's been wonderful to have you. Very educational for our team. I see lots of interesting and great questions. So thank you so much. And thank you to everybody who joined our call today. We appreciate your interest in the subject. Thank you, everybody. Have a great day.
And thank you very much, everyone, for inviting me today. Thank you.